There we go. Recording in progress. Wow, this is the first time I've heard a voice. She's never done a voice before. All right. Okay. I think with that, Karen, I think we're going to hand it over to you, and I'm going to get. Oh, and Daniel is here, and he has unmuted himself. Hello, Daniel. How are you?
Good, thanks. How are you?
Yeah, really, really good. Really, really good. And Karen, thank you very much for joining us today to introduce this paper. So I think when you're ready, please take it away.
Well, thank you, Grant and David and Marianna, for inviting me to participate. So this is the first Seabird session that I have done. So I'm happy to know a little bit more about this group, although I did see some messages passing by. But of course, everybody's so busy these days, we don't always take the time to do things that in the end are very rich and rewarding. But anyway, thank you very much. And I also want to thank you because it really gave me the chance to look at this paper. So entitled Extreme Phylopatry and Genetic Diversification at an Unprecedented Scale in the Seabird by Daniel Danckwert and his colleagues. And this paper, I really thought it was an excellent paper. So I'm really glad that I took the time to read it over carefully. Because it not only looks at seabirds, which I'm quite interested in, but it also looks at seabirds at Reunion Island, which I also am quite interested in. So it was a sort of took me back to Reunion Island and some of the work that I've done there. So anyway, I'm just going to summarize kind of briefly the paper. So we all know that in order to put into place effective conservation measures, quantifying things like genetic variation, structure, and gene flow is really an essential step because it enables us to understand how much diversity we have, what the risks of losing this diversity are, in particular in association with different populations. And it also enables us to get some idea of gene flow between populations, which of course is essential because we know that if a population starts to go locally extinct, it can be rescued by other populations if there's some gene flow. So it's really important, and particularly in seabirds and long-lived seabirds, to understand how much gene flow there are between these different populations. And we also know, based on some past work that we've done, that seabird populations can be more or less structured at different spatial scales. So although they can move as far as they want, when they want, in terms of reproduction, they tend to go back to the same places to breed. And so we could expect that there should be high gene flow between populations, but when we start to look at the genetic data, we see that this is often not the case. And in the past sort of 2 decades, we're really starting to get a lot more information about the different seabird species, how much structure they have in their populations, and what are the factors that are shaping this structure. And so in this study, the authors are looking at fine scale structure in a seabird that's endemic to Reunion Island, so the Barau's petrel, a medium-sized petrel. And they compare data from 2 colonies. And for a population genetic study, 2 colonies is not a lot. But for this species, there aren't that many colonies known. Daniel can correct me, but I think maybe these are the only 2 colonies that are really known. And these colonies are only separated by 5 kilometers. So this is really, really close, especially for a seabird. So this paper, they sample a really good number of individuals. So they've got over 100 in each of the 2 populations that they then genotype at 14 microsatellite markers. They carry out a suite of different analyses on these markers that are pretty standard, but very complete. So I'm pretty picky about these kind of things, and I really found nothing to criticize in terms of the treatment of the genetic data. And what they find in this study is that they have really obvious structure between these two geographically close populations. And so the structure in terms of a measure of population differentiation, so FST, is only 1%, which is relatively low, but it's significant. And what's particularly interesting in this data set is that when they carry out clustering analyses, so they either do that with discriminant analyses of principal components or with analyses in structure, is they find really clear indications of two distinct populations that correspond perfectly to the sampled populations. And more interestingly is when they look at the assignment levels of individuals. So the assignment levels are the probability when you take the multi-locus genotype of an individual, you can assign it to a particular population given the frequency of those different alleles in each population. And you can say what the probability an individual has of being from one population or another. And when they do this, they find really consistently that individuals are assigned to their population of origin. And there's no evidence of any migrants. So often in these assignment tests, we'll see sort of one individual that has kind of a lower probability of assignment than another. And this is really not the case. So here you have a really nice indication of structure between these two populations. And the other thing that they do in this study that I really liked is that they analyze additional morphological and banding data. And in terms of morphology, although this is going to be one of the questions I have for Daniel, they see some differences, but they're not very strong. And they're basically in morphometric measures, so basically size. But in terms of banding, the data is really consistent with the genetic study. So they see no evidence. They've never found any individual that's been banded in one of the populations in the other population. So these different lines of evidence really, really support the idea that these populations are quite distinct. And finally, the other thing that I wanted to highlight is that they use their microsatellite data to estimate population sizes and to look at whether there are any evidence of population bottlenecks in these populations. And in terms of population sizes, the estimates they get using the genetic data are relatively coherent with what the count data that they're seeing in the colonies. So I can't remember the exact numbers, but I think they're estimating over 1,000 individuals in the Piton des Neiges population, whereas they've got about 500 individuals in the Grand Benard population. And these seem to correspond relatively well with census data. So overall, the population genetic data is very, very coherent with what they're seeing and what they've been inferring based on other types of data. As I said, I really don't have any major criticisms of the way the data was handled. The data was very clean. It went through all of the normal steps of population and genetic study. And it was extremely well presented and clear. So if you're looking for an example to follow for carrying out your population genetic study on your favorite seabird species, I highly recommend this paper. It's very nice. In terms of the implications of this work, I think that it shows really that each population here is harboring a lot of novel diversity. One thing I didn't say is that they have a lot of private alleles in these populations. So that means even slight declines in the populations means they're going to lose genetic diversity. And so for conservation purposes, it means that each of these populations are very important to be maintained, and that they're not going to rescue the other. So if one of the two populations starts to go extinct, it's going to disappear, and that will be the end of it. And the other thing that I think is particularly interesting in this study, and another thing that I would like to discuss a little bit more with Daniel, is the idea of extreme phylopatry leading to population divergence and eventually to species divergence. So we see here that although differentiation isn't really strong, it's very, very clear, and it seems to have persisted for a long time. They don't see any sign of recent population bottlenecks. And so the question is at what point are we going to consider these two entities different species? Why don't we already consider them now? And I think this paper is a nice paper highlighting sort of the importance of phylopatry in generating species diversity in these very fragile species. So maybe I will start the discussion. That's all I wanted to say about it. That's all. Maybe I'll start the discussion with a couple of questions, and then everybody else can kind of take over. What surprised me in the analyses that was done was this absence of any evidence of a population bottleneck, and in particular, so the results found that the population seems to have been stable for several dozen or up to 5,000 generations, depending on which analyses they consider. So for a seabird that lives 30 years, this is a long, long time. And I was a bit surprised by this. And in particular, given the current threatened status of this species and the current impacts of humans and human-associated animals on these populations. So one thing that I didn't say is the Barau's petrel. Petrel is it's endemic to Reunion Island, and it's of conservation concern because of predation by cats and by rats, because it's breeding on the main island, and also due to light pollution. So a lot of young birds are lost when they come back towards the colony with the lights from the cities and towns. So they think the lights are calamar at sea, and they end up landing in the wrong spot, and then they can't take off again. So there's a system of rescue that's been put in place in Reunion Island to help these birds. So given these effects, at least these recent effects, I wondered why there was no sign of population bottleneck. And I wondered whether, Daniel, you could comment on this a bit.
Absolutely. Thank you so much for that wonderful introduction, Karen. That was really a nice summary of the paper and some of the different approaches that we used and the different findings that we obviously came up with at the end of things. So to comment on the bottleneck in slightly more detail without getting too in-depth in the analysis, we used 3 different analyses to try and identify genetic bottleneck within the species. One which has a fairly, informs us over a fairly low number of generations, one with a slightly higher range, and then the main method that we used was a coalescent approach based on an MCC modeling method. And like you said, this told us that the Barau's petrel population has been stable for roughly about 5,000 years, or 5,000 generations at least. And we know that the population size is roughly about 14,000 individuals at the moment. Now, as you mentioned, the main threats to the Barau's petrel are the predation of adults, chicks, and obviously their eggs up at the colonies by invasive mammals. We've got rats, mice, and even goats up there that are sort of having an impact on the habitats that these birds are breeding in. They're a very habitat-specific bird, which is quite key to our genetic results, I think, given the context of the mountainous areas in which they're breeding. But as far as the threats go, these are all extremely recent processes. So yes, they have had a fairly significant impact on the Barau's petrel population, but they're very recent threats that the species is facing, linked to man's colonization of Reunion Island, and then the subsequent population increase of us humans on sort of development on the island, particularly as far as the light-induced mortality is concerned. In saying that, it's also quite key to mention that there's been some incredible efforts by my colleagues on Reunion Island, as well as the SEO, which is the Society for the Conservation of Birds on Reunion Island, basically rescuing those grounded fledglings. And this has had a major reduction in the mortality that we've seen of these grounded fledglings. It exceeds 90% reduction in the number of birds that were being killed every year. So although the bird is facing some fairly large threats, they are relatively recent processes, and there are strong efforts to try and mitigate these as far as possible. So I don't think that is going to have had the time it requires to result in genetic bottlenecking in the species, at least not yet. It may well do so in the future, but at the moment, where things stand, the genetic seems pretty good within the species. Obviously, it's not great. Being an island endemic, we don't see as much diversity as we would expect in more widespread species, but it's good. And it isn't showing signs of reducing at the moment.
Okay, thanks a lot. I think that this is really an interesting point because I think I guess there are probably ways that we could estimate with current survival rates when we would expect to start to see a genetic effect.
Absolutely.
But with this kind of bird, it could be really interesting to try to do some modeling to see how many generations are required before you start to see a bottleneck given current survival rates. No, but yeah, I agree with you. I think that this is, I think that's something that we need to consider a little bit more when we're doing some of these genetic estimators, is to think about the time frame, and it should put us on an alert to what we might expect in the future.
Absolutely.
You mentioned this number of 14,000 estimated individuals, and I was curious, where does this come from, and where are these other populations?
So we, just to clarify those things, this point for those who may not be listening or who may not have been to Reunion Island, may not necessarily know the context in which the species breeds. Reunion Island is an incredibly mountainous island. The island is only about 50 kilometers in diameter. It's quite a sort of circular shaped island. But the highest peak on the island, Piton de Neige, which is where one of our study populations exceeds 3,000 meters above sea level. So we really are from the sort of tropical setting down at the coast. Once you get up to these study colonies, it becomes extremely cold at night. It's a very specific microhabitat these birds are breeding, and they're targeting this area in that it's got the optimal soil conditions that they require for their burrowing. Now, with that, many areas of Reunion Island are still totally inaccessible. We cannot get to them. So while we know of certain other colonies in which, or other areas in which the birds certainly seem to be breeding, the two colonies that we work in and the two colonies that we sampled are certainly the only accessible breeding colonies of the species on the island. Now, we estimate that the Piton de Neige population, which is the larger of the two studied populations, includes roughly about 800 pairs in total, so about 1,600 birds, of which about 250 nests are monitored on an annual basis. Grand-Benat is a slightly smaller colony. It's only about 400 pairs in total, and we are monitoring about 100 of those nests on an annual basis. We know of other areas that the birds are breeding. We just simply cannot get to them, but they seem to be fairly small colonies elsewhere in isolated pockets of the suitable habitat that the birds are targeting. My colleagues on Reunion Island have done some extensive radar surveys monitoring the number of birds that are flying in over the coast every evening. And that is the main indication of the population size. Obviously, then sort of extrapolating across to non-breeding birds that may not necessarily be visiting the island, as well as premature birds that aren't yet returning to the island to breed. They've also done some incredible work counting the number of vocalizations and using vocalizations at potential breeding sites to determine sizes of colonies and to see how the birds are distributed through these mountainous zones in which they are breeding.
Excellent. Thanks a lot. That's I was very curious about that. Another So just to clarify this, so do we know if they're on any other islands other than Reunion for now?
There are fossilized records of a petrel, very similar petrel by the looks of things, on Amsterdam Island. And there have been some nesting attempts on Mauritius, but at least birds displaying above Mauritius, but so far no nests have been found. And as such, the birds are considered to be a full breeding endemic to Reunion Island.
Okay. Just before I pass the questions over to other people, I was curious about the morphological differences that you found. So in the manuscript, you guys kind of play them down a little bit. They're significant, but you say that they're not really very different. But when you look at each measure, what you see is I think it's in the Piton de Neige population. They're just overall slightly bigger. Than the other population. I just wondered why you think that difference exists. Are the populations sharing the same resources? And
yes, so this is something that came basically at the end of the line. So this genetics is towards the end, if you will, of the various studies that we've done on Barau's petrel, at least the studies that my colleagues on Reunion have done. Patrick Pinet, who's a co-author on this paper, did some fairly extensive tracking work on Barau's petrels, looking at habitat use. And for my master's, I then looked at a bit of a trophic study, looking at how the birds are sort of using the different habitats that they are occupying through different periods of the year. And there seems to be virtually no difference at all in terms of the habitat use and resource acquisition between these two colonies. The birds are using very clearly defined areas of the Indian Ocean during both the breeding and the non-breeding period. But then quite interestingly, the breeding period is split into 4 distinct periods. The pre-laying exodus, the sort of egg-laying period, incubation, and then chick rearing. And the birds are using completely different sections of the southwestern Indian Ocean during those different periods. They first start off foraging quite near the South African coast, and then they move closer and closer back to Reunion Island. And that seems to be consistent between the two breeding colonies. They track birds from both breeding colonies, and they are using the same areas throughout the year. So the morphometrics was essentially one of those final things that we were looking at in terms of something that hadn't yet necessarily been done, and essentially just to see if the genetic differences had sort of translated into any kind of phenotypic differences. If there was any sort of difference in size, we know that plumage-wise, the birds are pretty much exact between the two breeding colonies. And although the wing length seems to come out as driving a difference between the driving what difference exists between the two breeding colonies, it very definitely was sort of non-significant, and there wasn't much to it, but we felt it served a very important place in the paper as something to establish just how fine scale this genetic difference is. You mentioned that at what point will these two genetic populations become separate species? It's important to realize that at the moment we are looking at the population level. This hasn't even reached subspecies level. Whether or not they will ever diverge sufficiently to become separate species, I don't know if they will. It obviously requires sufficient sort of external stimulus to push those speciation events apart. So the birds certainly have unique, or the two populations certainly have unique genetic material in them, but this is still at an extremely fine scale, and it just highlights in the greater scheme of things how where you have populations separated among different islands, for example, how easily those speciation events can happen.
Well, I think that's interesting, but I'm curious about why there's no consideration of a subspecies status or this kind of thing at this stage, because of course it depends on what you use as your reference point for defining species. But here you seem to have two populations that are genetically distinct, that have no current evidence of gene flow, that are morphologically distinct. And given that they're using the same resources, they're potentially, this difference is potentially genetically based. And I'm curious, do you have data from, because you don't mention anything in the paper about other markers, so more conserved markers, for example, mitochondrial markers. I'm sure this data probably exists somewhere, but that might give you a little more perspective on this, because microsats are very recent, of course, and they work well for both populations. So it suggests that any divergences are indeed very recent. But where do we draw the line between these things is not at all obvious. Do you have any data from complementary markers that can give you a little bit more of a historical perspective on when these two populations diverged?
Not as yet. I do believe there has sort of been some preliminary investigation into using mitochondrial markers to try and see whether or not these populations are diverging at those scales yet. But given our results with the microsatellite information, I would be quite confident that the difference we observed between these two populations using microsatellites would not yet be reflected in mitochondrial DNA. Should we perform the study in another couple 100,000 years, it may well be the case, but at the moment I don't think it has had the time nor the stimulus to result in such a divergence. This is still looking extremely fine scale at that population level rather than subspecies.
Okay, thanks a lot. Maybe leave it over to Grant and David and Marianna if you want.
All right, let's start with you, David.
Yeah, well first of all, thanks so much, Karen, for introducing the paper, and you brought exactly the expertise we were missing. So thanks a lot for joining. And I think we skipped that. We skipped saying that you're senior researcher with CNRS in Montpellier, and I encourage our young colleagues to check on the great work you're doing there. And thanks also to Daniel for the very thorough answers. And actually, you answered my first question, which would have been about the at-sea space use of birds from the two colonies. Because I was thinking, if there's such a difference between the two colonies, this may be mirrored in their at-sea use. There might be exclusive foraging areas, which we very often find in neighboring seabird colonies, but I guess it's less the case in petrels, and it's more often the case maybe in alcids or sulids or cormorants. But I was getting excited about this idea that as we see asymmetries building up in the way birds use the space at sea, this may contribute to the fact that then they become genetically distinct. And that would be, I think, a very neat way to go all the way from individual behavior at sea to then building up a sort of a foraging culture, and then excluding foraging zones, and then which would mean that even neighboring colonies become distinct because birds never mix. But actually, in your system, that's not the case, because they use the same areas at sea. But so to go on to a proper question, I was wondering what gave you or your colleagues the hint for doing that genetic study. was there already a suspicion that there might be this split between the two colonies?
I'm gonna, if you wouldn't mind, I'm gonna answer that question in two parts. The first is that this formed a chapter of my PhD, looking at scale-specific processes that underlie or that determine the ways in which seabird populations are structured, ranging from an island endemic species that is distributed in a non-continuous manner. We've got isolated populations in a sort of geographical, topographical sense, all the way through to, I've studied 3 different species. It was the Barau's petrel, the red-footed booby, and then the sooty tern. And it was quite interesting to see how the different species are genetically structured across the various different scales. I mean, where we have the sooty tern, even using microsatellite information, we weren't able to find any distinction between or among any of the colonies that we sampled across the entire Indian Ocean, as well as a sort of an outgroup colony from the Pacific, which shows that using the microsatellite information, the sooty terns are moving moving among colonies, not just at the scale of the Indian Ocean, but at the scale of the entire Indo-Pacific. We're confident we weren't able to include any Atlantic samples from that. They have studied the same thing using mitochondrial DNA, and they found that the Atlantic sooty tern population is quite distinct from the Indo-Pacific population, and we were interested to see if that could be reflected in microsatellite information, or if the microsatellites were able to detect the occasional spillover around South Africa or maybe across Panama or something like that. So the entire PhD study formed an analysis of scale-specific processes ranging from these extremely fine scales in the Barau's petrel, where we weren't expecting to find a difference, all the way up to the sooty tern, where for different reasons we weren't expecting to find a difference, but it was interesting to see how seabird populations are structured like that. Then, in terms of where the study originates, the study formed a part of the Life Plus Petrels project, which ran between the years of 2014 and 2020. And this was supported by the European Life Program. And it was a massive, massive study on the 2 endemic petrels of Reunion Island, the Barau's petrel and the Mascarene petrel. And it was essentially the overall objective of this program was to try and halt the declines in the 2 species by providing all the information we needed to come up with the strongest action plan we could to prevent further declines in both species. So that incorporated several different studies, including this genetic study, but it was a huge, huge research program on the two petrels. We had the money to do it, and we, although we weren't necessarily expecting to find this level of structure, we had the intention to sort of describe genetic diversity within the 2 populations to see how they connected. And I still remember vividly the day we ran that structure simulation, which is the key result of the paper. Sort of, I saw the results of the simulation. I said, no, this can't be right. So I ran over to my colleague's office, and we sort of sat down for another 2 hours, reran the simulation. The result popped up, and all of us were sort of flabbergasted as to what these birds were doing. So certainly an incredibly exciting and unexpected result, but it came from sort of a general analysis that we had intended to do on the birds.
Okay, yeah, thanks a lot. Well, I have one more follow-up questions, but first maybe we should go around and see if folks have other questions.
Why don't we, we've got a question down in the chat, and then we can pop over to Marianna. So the question down in the chat is from Philippe, who asks, what about sampling the birds, 1 to 2% of the estimated population inside of the colonies? Could birds be sampled through all of each colony, or are they located close together due to difficulty of access? I'm not sure.
Sorry. So many of the breeding colonies are, I mean, I wish I had an image of Reunion Island to show you, just to show you how mountainous the central parts of the island are. And many of the areas that these birds are using are just completely difficult to access. The Grambena colony, a certain section of the Grambena colony requires a fair bit of rope work to access. And you've got to be strapped in a harness just to make sure that you're not going to fall off a several 100-meter high cliff. So really difficult access in some of these sites. It requires a very long hike to get to the two breeding colonies that we study, and that says a lot for the colonies that we aren't able to access. In saying that, Karen mentioned in the beginning that one of the main threats that this bird is facing is the light-induced mortality of fledglings. So every year in about April, when these birds start leaving the colonies, they leave the colonies at night. As they fly down from the mountainous sections of the island, they've got to run the gauntlet of the cities and towns around the coast. And we're seeing hundreds of birds every year grounded that my colleagues on Reunion have had to initiate quite stringent rescue programs to save these birds from an ultimate fate. What they are now doing is sampling those chicks. So those chicks are essentially non-random. They are coming from colonies throughout the island, not necessarily just the two colonies that we have direct access to. And we're going to see if the patterns are the same genetic patterns are evident in those populations, and how some of these other colonies might influence what the findings of our study, obviously gaining access to those birds through the fledglings.
Mm-hmm. Cool. All right. Well, while you're mentioning that, I quickly Googled a picture of Reunion Island, so I'll quickly share that just so people are aware. You can take a look here. This is an aerial view. You can see how mountainous and steep this island is. And yeah, I can imagine being harnessed up would be necessary in a lot of these colonies.
So those two peaks that you can see right in the center of the island, those two highest peaks, the one you have your mouse on at the moment is Petrel, and I believe the one to the right of that is Grambena. Yes, that I think is Grambena. So extremely mountainous. You're going probably, well, anything between about 2,600 and 2,400 and 2,800 meters above sea level. So really high up, really quite tricky terrain to work in, but we typically go up for a couple of days and actually stay in the colonies to do the work, because it is quite a trek in and out every time.
Yeah, I can imagine. So how do you camp? I assume there are some campsites up there, or is it, how do you set up when you're up at the top?
So Piton de Neige, we have a sort of a formal campsite. It's quite rustic, that you carry your tents up. A lot of the food and that is flown up with a helicopter in advance, so we don't have to carry all of that up. Getting to the Piton de Neige colony is really quite difficult and requires some rope work and all of that. Grambena is a slightly easier hike to get to the colony. This was the first study colony that we were working on the Barau's Petrel. What we have there is essentially a shipping container that has been flown up to the colony with a helicopter. It's cool. Strapped onto the side of the mountain. It's really spectacular up there. It's got some sort of makeshift bunks in it. It's got a sort of provision of food, and the roof of the little container is adapted to collecting rainwater, so you don't have to carry all that much water up with you. So at the Grambenard colony, we at least have a little bit more of a base to base ourselves, but we'll typically go up for 3 or 4 days at a time, get the work. It takes pretty much a full day to get there and a full day to get back. And we'll be up at the colony to do what work we need to do and then sort of hike back down.
Mm-hmm. And I always say that seabird biologists are mental. All right, Marianna, do you have any questions?
I am super impressed. I also had, hi Daniel, nice to meet you, first of all. I also had the question, how, like, what makes a study site actually accessible? Wow, mind-blowing, fantastic. So I have a very general question. So you also mentioned the presence of another endemic species on the island. So if you can tell us what you know about it, or if you are also like planning to make a similar study on it.
Perfect. So mascarene petrel is one of the world's rarest birds. Until recently, we were estimating a population of roughly 50 mature individuals. So this bird is critically endangered. The breeding colonies were only discovered within the last sort of 5 or 6 years using those radar surveys that I mentioned earlier, that they went out into the island using radar, monitoring where the birds were going to land, and then flying into those areas with helicopters to see if you could actually find colonies. Hadaram Shirahai then came to Reunion Island and he did some at-sea surveys and actually managed to find a fair number of mascarene petrels out at sea, which sort of cast a little bit of doubt as to how many birds, how many mascarene petrels there actually are. But what is clear is the bird is in an extremely critical state and is in urgent need of conservation action to try and sort of prevent the bird going from extinct. So they managed to find, I believe it's now 2 or 3 breeding colonies on Reunion Island, only a handful of pairs in each. And they've managed to get a sample from the breeding colonies as well as grounded birds every now and then to do a similar genetic study looking at so they haven't found the same level of population structure as we have in the Barau's petrel. Obviously, bearing in mind it is an extremely low sample size, but I believe there is some evidence of genetic bottlenecking in that species. I wasn't directly involved with that, so I am sort of remembering from the few discussions I've had with my colleagues about it. But the species had fairly unexpectedly high genetic structure, which was intriguing. But the conservation efforts on that bird are continuing. They are still trying to find more colonies and to do as much work as we can to learn as much about the birds as possible.
Yeah. And is the same, like they are threatened by the same factors as the other species, or also like at sea?
Indeed. So they have now done some tracking work on the mascarene petrels. The results are extremely preliminary. So the work is sort of still coming through the grapevine, if you will. But they're nesting in quite similar habitat, also up in the mountains of Reunion. And one would expect that the threats are very much the same, that it's going to be your predation by invasive mammals at the breeding colonies, light-induced mortality of fledglings as they leave the nest, and then there's obviously going to be an assessment of at-sea threats and things like that. The genetic results of the mascarene petrel are actually published. It's quite a small paper, but published by Laurence Humor, who is one of the co-authors on the paper on the Barau's petrel. Just the sort of preliminary investigation with the few individuals that they have been able to get their hands on.
Wow. But so just out of curiosity, I don't really, I'm not an expert in this field. So do you then expect to have inbreeding into the mascarene petrel or not?
Yes, so there are concerns of inbreeding depression going forward, and that would be something we would need to watch out for as something that could result in genetic depression and potentially reduce the bird's resilience and all of that.
Great, thank you.
Great. Thanks, Marianna. Do we have any questions from the audience before we ask some more? Nobody's raising their hand. Nobody's. Oh, wait. Jonas has a question.
Hi, Daniel. This was so interesting to hear. I was thinking along the same lines as David a little bit on kind of, I mean, clearly there is no like niche separation at sea or anything like that. But I was wondering, are there other ecological factors you could think of that would prevent any mixing between those colonies? Like, for example, is there very like fierce competition on nest sites or anything like that did you experience in the field?
Barau petrel is quite a habitat-specific species in terms of its nesting requirements. It targets this high-altitude elfin forest where soil conditions in particular are suitable for nest construction. Now, we do discuss this to a limited degree in the paper. Obviously, word counts and all of that limited how much depth we could deal with this. But the Piton de Neige colony is situated between it averages at about 2,400 meters above sea level, whereas the colony that we're working on Grand Benat, is sort of at the upper altitudinal limit of the species. And there is quite a marked habitat difference between the two breeding colonies, where Grand Benat, the habitat on the ground is quite rocky and there's less access to suitable soil conditions for nest sites. We see much lower nest density in Grand Benat relative to Piton de Neige, where the habitat conditions are so optimized for breeding in the species. But that doesn't really reflect too much in the genetics, in that the lower parts of the Grand Benat colony where we're able to access most of our birds is sort of at the upper limit of the Piton de Neige colony. So the lower down you get at Grand Benat, the more optimal those habitat conditions become, and that's where the majority of those birds are breeding. But this high altitude habitat that the birds are occupying is distributed only in those very uppermost mountain sections of Reunion Island. And so the distribution of the habitat, combined with the tendency of birds to return to the natal colony, so that high natal phylopatry, seems to have been what's driven this genetic difference between the two populations. That it's a combination of the birds' habitat selectivity and the distribution of said habitat, as well as the tendency of birds to go back to their natal breeding site.
Very cool. Thanks, Jonas. Any other questions from the audience?
I actually had no.
Go for it, Karen. No. Go for it, Karen. Ask away.
Sorry. I keep getting more and more questions. But Daniel, I was wondering in relation to that, whether you have any information about where pair formation occurs. Are they forming at sea? Are they forming pairs in the colony? Do you have any data about that?
What's fascinating about Barau's petrel, it's unique in a number of senses. So Barau's petrel is one of the highest nesting seabird species in the world in terms of the altitude that it's nesting at. And the birds have a tendency, or a lot of the birds, not all of them, which we've now seen from the radar surveys, a lot of the birds actually arrive back on Reunion Island during the day when they come to provision their chicks, or when they do incubation changeovers and such. And that's quite unusual for a gadfly petrel. Most gadfly petrels arrive back at the breeding sites at night, and all of the activity happens at the colonies at night. So sitting up at the colonies at dusk during the peak breeding season, you see these clouds of birds coming up through the big valleys that they use to sort of gain altitude. And then there's a lot of displaying and calling that happens above the breeding sites. And then they sort of all drop down into the colonies as soon as it gets dark, and they go to find their nests or provision chicks or anything like that. So the birds certainly seem to be displaying together when they come up to, they essentially display over the big valley that separates the two peaks, and then they move closer and closer to the colonies before they eventually drop down into the breeding colonies. So it's interesting to see how they are displaying together, but yet there's still this strong sort of homing into that natal colony. One of the next things that we hope to do on the species is to try and quantify the smell of the birds to see if there may be some sort of olfactory difference. Anyone who's worked on a petrel or had, well, any seabird at all will know how much some of these birds stink because of the oils that they put out of their feathers and all of that. So we are interested to see if there is any difference in smell between the two birds. And the theory goes that these birds may be incredibly reliant on their olfactory senses, given their nocturnal behavior at the breeding site. So to see if that could be another potential cause for the separation between the two colonies. But that method isn't properly developed yet, so it will take some sort of clever thinking to figure out how one would be able to quantify the difference in smell between birds.
Super cool.
Okay, super interesting. Can I ask one more question? And then I promise I'll stop.
That's fine. We'll get a question for you, Karen, and then we got one more question in the chat, and then we'll call it quits.
Okay, I just wanted to ask whether you guys know anything yet about what the young birds are doing. So first reproduction is around 6 years. These young birds are never seen in the other colonies. Do we know anything? Are they staying at sea and they just come back when they start to reproduce, or are they hanging out in the colony? Or
we've got very little evidence to suggest, based on the banding efforts, we've got very little evidence to suggest that the birds are coming back and sort of prospecting in the colonies before they reach sexual maturity, which would suggest that they spend a lot of their time out at sea. In saying that, our efforts to capture banding, to recapture banded birds has been a little bit biased towards sort of the breeding, the chick rearing period, and not necessarily when adults are coming to do their prospecting. So I mentioned the birds have a fairly complex breeding strategy. They arrive back in about September, October. The birds display above the colonies for a couple of days. They find their nests. There's a bit of pair bonding going on. And then they go out on what we call the pre-laying exodus. So at that time, there's a clear separation between the male and the female bird. The males tend to fly a lot further. They're foraging closer to South Africa, whereas the females are staying immediately to the south of Madagascar. They then both return at a very similar time to Reunion Island in about November. So that period is about a month where they're not at the breeding colonies. The female will lay the egg and the male assumes the first incubation shift, which is always the longest of the incubation shifts, and the female immediately goes back out to sea. And then they start the rotation through the breeding period into the chick rearing period. Most of our efforts within the colonies are once the egg is laid and during the chick rearing period. And then there's a big banding effort just before fledgling go in, and we try and bring as many of the chicks as possible from that particular season. So we would need to spend more time in the colonies during that initial period where birds are coming in to display, to see if any of those immature birds are coming in with the adults. But it is unlikely, and there have now been efforts to track the immature birds in addition to the adults to see where they are going once they leave. And it does seem to be quite different to what the adults are doing. So the chicks seem to be heading north, where the adults immediately fly east from Reunion to the 90-degree ridge in the Indian Ocean. And they spend the non-breeding period out over that, where some of the chicks are going, the chicks that we've been able to track are headed straight north up towards Somalia and the Seychelles.
Cool. All right. Thanks for that. All right, there's actually 3 questions and one down in the chat here. So we might just pick one of these 3 and call it quits for now, just so we don't go too far into everybody's days. So we've got 3 questions. Is there a difference between? Okay, let's take the question that kind of leads from what you guys were just talking about, which is basically, is there differences in breeding success between the 2 colonies?
Ever so slight differences. One would expect that, just in that it is slightly different habitat. It's slightly different incidence of predation by invasive mammals and things like that, but it's not a striking difference at all. Breeding success does seem to be quite good, especially now that my colleagues on Reunion are doing such intense efforts to control the cats and the rats at the breeding site. Yeah.
Well, that's the second question that they asked down there, actually, was whether or not there was any conservation measures to avoid mammalian predation. So you've answered 2 questions in one go. Well done. All right, I think with that we can probably end our discussion and stop recording. And so just quickly, thank you for joining us today, and thanks for a really cool paper, Daniel. Obviously this has generated some really great discussion today, and we've managed to fill a whole hour with one paper. So that's really fantastic. And Karen, thank you very much for introducing the paper and for asking some very well-informed questions. It's very great to have that. All right, and I think with that I'll stop recording, and then